The overall goal of the following experiment is to fabricate and characterize conjugate nanobiological systems interfacing solid nanostructured substrates with immobilized biomolecules. This is achieved by utilizing ultra-high-resolution electron beam lithography, or EBL, to obtain arrays of tiny metallic nanostructures on fused silica supports. As a second step, the substrates are placed in solution and biomolecules are immobilized on their surfaces, producing the nanobiological interfaces. Next, surface-enhanced Raman Spectroscopy, or SIRS, is employed in order to probe the biological molecules at the interfaces. The resulting SIRS spectra exhibit Raman modes characteristic of the particular molecules, with intensities strongly dependent on the substrate design.
The main advantage of our approach over existing techniques, like infrared spectroscopy, is that the sensitivity can be greatly enhanced due to surface plasmon resonance within the metal nanostructures. Our approach creates highly selective surfaces that can be readily controlled using the combination of molecular functionalization and electron beam lithography.
This method can answer key questions across multiple fields, from exploration research in structural biology or surface-immobilized biopolymers to the development of various devices involving nanobiological interfaces.
These methods can provide insights into the structural properties of biomolecules, with implications extending towards medical diagnosis or environmental detection.
Generally, individuals new to this method can struggle as it involves aspects of nanofabrication, surface functionalization, biochemistry, and spectroscopy.